Hello everyone, this is Utabiso Mhlongo. Some know me as Mr. Ray. I'm one of the tutors at Limitless Academy. I'm a life science tutor and I tutor grade 11 and 12 life sciences. We are doing these videos to help during this time, so we are going to do revisions and also do new topics which should be done at the second term. If you feel that you need to ask any questions, the numbers on the screen will help you and will get you in contact with the tutor that you need to ask questions to. And I'm also willing to help anyone who comes to contact with me and ask me questions based only on life sciences, nothing more, only on life sciences. So that's why I'm urging you to ask questions. If during my explanation or during my tutoring sessions you did not understand something, you are allowed to ask me anything via WhatsApp, or you can call me, or you can also find us on our Instagram page, Facebook page, and yeah, that will be it. So today for life sciences, grade 12, I'm starting with grade 12, and we are going to be talking about, we're going to do a revision on the first topic for the first term, which is DNA, the code of life. So when we are talking about DNA, the code of life, we know the word code. We all know the word code. When we're talking about code, it means it's something, it gives instructions to what should be done or what can be done with a certain sequence of things. So we are talking about DNA as the code of life. But before we go into DNA as the code of life, we need to know what makes DNA. What group does DNA fall under? So DNA falls under a very marvelous group which is called nucleic acids. So when we're talking about nucleic acids, we are talking not only about DNA, but we are talking about two nucleic acids. We have RNA and DNA. When we are talking about RNA, it's ribonucleic acid, as you say, ribonucleic acid. And then this is the nucleic acid part. And then we also have deoxyribonucleic acid. This is the nucleic acid part. So both RNA and DNA are nucleic acids. I'm not going to dive deep into these topics, especially the first 10 topics. I'm just going to do a light revision on them, and then you can ask me questions later. So when we are talking about nucleic acids, as you all know that whenever something is there, there are things that lead to the formation of a thing. It's like when you are cooking, you need ingredients so that you can make your food. When you are building a house, you need bricks, you need cement, you need sand, you need whatever that you need so that you can make a house. So do we, when we are talking about nucleic acids, we have to have things that make nucleic acids, which we call them nucleotides. They are called monomers. Monomers, which are the smallest building blocks of nucleic acids. So when we are talking about nucleotides, we are talking about the things that actually lead to having what we call nucleic acids. Okay? Okay, nucleic acids, we need nucleotides. What are nucleotides made up of? So is the structure of a nucleotide. It's made up of three components. We have a nitrogenous base, we have a sugar portion, we have a phosphate portion. In some books, they say it's a phosphate ion, which is ION. So this is what we need so that we can have what? Nucleic acids. We need to have nucleotides, which nucleotides are made up of nitrogenous bases, phosphate, sugar portion, phosphate portion, or phosphate ion. Okay, so I've already said that under nucleic acid you have RNA and DNA. When we are combining both RNA and DNA without actually separating them, we have 5 nitrogenous bases. Between RNA and DNA, we have 5 nitrogenous bases, which are adenine, guanine, cytosine, thymine, and uracil. But when we separate them, when we are talking about DNA only, we have 4 nucleic acids for DNA, which is deoxyribonucleic acid. When we are talking about ribonucleic acid, we have 4 only. But when we are combining, talking about them in combination, we have 5 nitrogenous bases. Okay. So now let's talk about DNA. When we are talking about DNA, we are talking about deoxyribonucleic acid. So the full name for DNA is what? Is deoxyribonucleic acid. Okay, so we have 3 types of DNA. We have, when we're talking about DNA, we talk about the location of DNA. We call DNA using its location. So DNA is found in the nucleus, which we call nuclear DNA. And then we have another one, which is the mitochondrial DNA, which is found in the mitochondria. And then we also have chloroplastic DNA, which is found in the chloroplast. Since all living organisms, all living cells have a nucleus and a mitochondria, but then a chloroplast is only found in plants. So a nucleus is found in animals and plant cells, and mitochondria is found in both animals and plant cells. But a chloroplast is only found in plant cells. So these type of DNA you only find in plant cells. Okay. So these are the locations of DNA. We find it in the nucleus, and then we call that one nuclear DNA. And then we have mitochondrial DNA, which we have mitochondrial DNA, which we find in the mitochondria, and then we have chloroplastic DNA, which we find in the chloroplast. And then we have the function. What is the function of DNA? What does it do? What makes DNA so special that we have to actually have a chapter talking about DNA? So the first function of DNA is what? The first function of DNA. Okay, the books that I'm using this green book, which is Understanding Life Sciences, which is the 3rd edition, and I also have my other study guide, which is X-Kit Achiever, Grade 12 Life Sciences. They are both good books. The reason why I love to use different books, it's because they explain in different ways. And then when they explain in different ways, I am able to then find the one that I think will be easier for you to understand, and then I use that book. So I usually use this book, and also another study guide is the Mind the Gap study guide. You can actually use that one again. There are many other study guides that you can use for explaining or learning life science. Okay, so now we are going to talk about the functions of DNA. So when we are talking about the functions of DNA, I'm going to focus on 2 functions, on 2 main functions of DNA. Not that there aren't any other functions, but there are 2 functions that I like us to focus on. Which is the first one, it's nuclear DNA controls the synthesis of proteins. When we're talking about controlling the synthesis of proteins, it means it controls the manufacturing of proteins. So DNA, nuclear DNA, when we're talking about nuclear DNA, as I've explained before, we have DNA esitola in nucleus, in nuclear DNA. Layer DNA iyona enzyme which controls in protein synthesis. So ukwenziwa kwama proteins kucontrolwa in nuclear DNA. Okay, so that is the first function, which is in nuclear DNA. controls the manufacture of proteins. That is the first one. And then again, the second function for DNA, what does it do? Nuclear DNA also transmits hereditary characteristics from parents to offspring. So nuclear DNA, again, what does it do? It controls the transmitting, or how can I say this? We are what we are because of our parents, right? So I am short because I inherited the gene for being short from my parents. I have a dark skin, I have wrinkled hair, I have whatever characteristics that appear on my phenotype or on my physical appearance. Those things, be it my physical appearance or my internal structures or whatever that I am, that is because of my parents. So DNA actually is able to transmit those hereditary characteristics. So as we know that a human has about 46 chromosomes. 46 chromosomes are from the maternal and paternal side. So when they combine, they form a human being, one person. So that person is as a result of the fusion of DNA from both parents. So DNA is passed down from existing cells to new cells during reproduction. In this way, the characteristics of parents are passed on onto their offspring. So those characteristics that you have when you like eating their that is because some characteristics have been transmitted from your parents to you. So DNA is responsible for that, for those characteristics that have been transmitted to you from your parents. So it transmits, again, it's nuclear DNA, transmits the hereditary characteristics from parents to offspring. Okay, so again, it's nuclear DNA that is responsible for the transmitting of hereditary characteristics from parents to offspring. So now, we have talked about the function, we have talked about the location, but we haven't talked about the structure of DNA. We know that DNA is one of the nucleic acids. We know that DNA, for it to be part of the nucleic acid, there are monomers that make nucleic acid, which are nitrogenous bases, a sugar portion, and a phosphate portion, or ion. Some books say ion, some books say a sugar portion, a phosphate ion. Some books say phosphate ion, some books say phosphate portion. So now let's look at the structure of DNA. When we are talking about the structure, how do you differentiate this is DNA and this is not DNA? So that is what we are going into now. So there are some topics that are under the DNA topic that I'm not going to talk about because right now we are dealing with revision. I'm not actually reteaching the whole topic. So if there is something you do not understand during term 1, feel free to contact me as I have said before. So we are just going to deal with the structure. Because of time, we are going to talk about the structure, and then I'm going to jump into RNA, and then I'm going to talk about the function and location and structure of RNA and also protein synthesis. But I'm not going to talk about DNA, we are just going to finish with the structure. Okay. So when we're talking about the structure of DNA, the first thing we need to know, which I've explained, we know that it's a nucleic acid, and we know that a nucleic acid is made of monomers called nucleotides, and then we know that nucleotides have three components that make a nucleotide, which are a phosphate portion, a nitrogenous base, and a sugar portion. So now we have to know the type of sugar that is found, the type of sugar in DNA. It's called a deoxyribose sugar. So this is the type of sugar that is found in DNA. It's a deoxyribose sugar. Not deoxyribonucleic. No. Once you say deoxyribonucleic, that means you are writing the full name of DNA. But when we are talking about the type of sugar, it's called deoxyribose sugar. So that is the first part, the type of sugar that is found there. And then we have nitrogenous bases. When we are talking about nitrogenous bases, as I did explain before, that when we are talking in combination, when we are talking about nucleic acids, we have about 5 nitrogenous bases between RNA and DNA. Kodwa uma ngabe sikhuluma nge-DNA, ngidila ne-DNA ngingayifaki i-RNA, sinama-nitrogenous bases amangaki? Awu-4. So lama-nitrogenous bases ayahlukaniswa into 2 groups. We have purines. And then mase siphinda siba nama-pyrimidines. Pyrimidines, whichever way you pronounce it, as long as you're gonna write the correct spelling, which I'm not good at sometimes. My learners will tell you that. Okay, so when you're talking about purines, I'm talking about adenine and guanine. And then when you're talking about pyrimidines, we are talking about cytosine and thymine. So what you should know again, DNA is a double-stranded molecule. It's double-stranded. It's double-stranded. When I'm saying it's double-stranded, that means it's not made of one single strand like this, but it is made of 2 strands. And its natural shape is a helix. The natural shape of DNA is a helix. When you're talking about a helix, I'm talking about something that is like a spring like that. When we look at each spring, this is on your bed, that thingy, that is how it is shaped. So it is a double helix. But when we're doing DNA replication, whereby it's another process whereby DNA replicates, it makes an exact copy of itself. It needs to move from a double helix and form a leather-like shape. So when we are talking about DNA, since it's a double strand, each strand is made of nucleotides. Nucleotides. And then within each nucleotide, let's say these are your nitrogenous bases. These are your nitrogenous bases. Okay, so let's say as we have separated them, we have adenine, guanine, cytosine, and thymine. We have adenine, cytosine, guanine, thymine. Okay, let me just do a short strand, not the whole, not all of them. I'm just going to do a short one. Okay, let me, so DNA, once we are looking at, since we are looking at it like this, these are just 2 strands standing on their own. But remember what I said, that DNA is a double strand. So when they are like this, they are just single strands. There is nothing holding them together. So if they are like this, they are able to flow apart from each other. But because there is another thing that we find in DNA that we do not find in RNA, which is called the hydrogen, the weak hydrogen bonds. It's not just hydrogen bonds, but it's weak hydrogen bonds. The reason why they are weak hydrogen bonds is because DNA needs to break when we are having DNA replication, and also DNA needs to break when we are having protein synthesis. So if these bonds are too strong, those processes might not happen, or they might take too long to happen, which our bodies need them to happen like fast. So DNA also has some things called weak hydrogen bonds, holding the strands together. Okay, so we have these weak hydrogen bonds that hold the nitrogenous bases together. Again, for it to be a double strand, that means the nitrogenous bases need to pair with each other. When we are saying they need to pair, they need to work in pairs. They need to be, they are partners in crime, if I can say that. So purines pair with primarides. So another easy way to understand them is just say at GC. By saying at GC, we are saying adenine pairs off with thymine, and then guanine pairs off with cytosine. So this is how they pair with each other. So adenine with thymine, guanine with cytosine. That is how they pair. So they form that double strand. Okay. Okay, so we're talking about weak hydrogen bonds. That DNA, since it's double-stranded, that means it's two strands joined together by something. Nitrogenous bases pair, they pair with each other. So purines pair with pyrimidines. We have adenine paired with thymine, and then we have guanine paired with cytosine. And I'll get this research at GC. That means adenine, thymine, guanine, cytosine. Since it's double-stranded, there has to be something that holds the nitrogenous bases together. And what is that? It's the weak hydrogen bonds. You cannot just say hydrogen bonds, but you have to say weak hydrogen bonds because they are weak. Okay, I have explained it. So now I'm gonna just do a brief highlight on DNA replication now. Yes. So we do know the type of sugar, we do know the shape of DNA, we do know the bonds and the complementary bases. We do know that which is adenine, thymine, guanine, and cytosine. So now I'm just going to do a brief explanation about DNA replication. I'm not going to go deep into it since we are doing revision, but at a later stage I'm going to try and find past papers so we can work with past papers and answer past papers. So because of the time constraint, I'm just trying to be as quick as possible. So when we are talking about DNA replication, this is a process whereby DNA makes an exact copy of itself. So DNA is making an exact copy of itself when it does replication. When does DNA replication? We do remember that we have in grade 10 we did mitosis, which is the cell division of somatic cells, the body cells. And then again this year, grade 12. In grade 12 you did division, which is called meiosis. So DNA replication occurs in interphase, in both divisions. That is mitosis, it occurs where? In interphase. And in meiosis, it occurs in interphase before meiosis. So it's interphase and then meiosis 1 and then you continue. So it's whereby DNA is making an exact copy of it, so that each and every chromosome may have the same DNA. So that as the end result, all chromosomes have the same number of DNA, the same DNA, they have the same genetic information that is needed by the cell. So let's go to RNA. RNA. So when we are talking about RNA is rather very simple. So in RNA, when we are talking about RNA, we are talking about ribonucleic acid. Ribonucleic. It's ribonucleic acid. The type of sugar that is found in RNA, it's called ribose sugar. It's called ribose sugar. Do not confuse the two. In DNA we have deoxyribose sugar, but in RNA we have ribose sugar. And then, remember when I said the nitrogenous bases between DNA and RNA, when we're talking about all of them, there's 5. Now we're going to see where the 5th one comes in. So we have nitrogenous bases. Right here we have adenine, guanine, cytosine. But then, instead of having thymine, we add what? We add another one, which is uracil. So now you can see you have 1, 2, 3, 4, and then you have 5. So we have adenine, guanine, cytosine, and uracil. Again, RNA, unlike DNA, is single-stranded. It's single-stranded and it is shorter than DNA. DNA is a long strand, but RNA is a very short strand, shorter compared to DNA. And it is single-stranded, which goes to tell you that if it is single-stranded, it does not have weak hydrogen bonds. It does not have any bonds. Why? Because it is single-stranded. You cannot have a single strand and then tell me you're going to have bonds at the same time. It does not have bonds because it is single-stranded. Now, the function of RNA. What is the function of RNA? There is only one major function, one important function of RNA, which is the simplest one, which I think you should all remember. They all 3 types of RNA, because there are 3 types of RNA. Okay, let me work. So the sugar is ribose sugar. And then nitrogenous bases, you have adenine, guanine, cytosine, and uracil. And then you have 3 types of RNA. We have messenger RNA and transfer RNA, and also ribosomal RNA. These are the 3 RNAs that we have. You have messenger RNA, transfer RNA, and ribosomal RNA. Those are the 3 RNAs. And all three of them, they play an important role in protein synthesis. So they all play an important role in protein synthesis. Without these, there is no protein synthesis. But where do we get these RNAs? Remember the function that I mentioned earlier on DNA when I said it controls the protein synthesis. Remember when I said this? Now, how does it control protein synthesis? What is it that DNA has that allows it to be able to control protein synthesis? Whilst protein synthesis is made from another nucleic acid, why is DNA so powerful? How is it? How does it come about? Okay, so DNA, when we are talking about, the reason why I skipped DNA replication is because I was going to do a little bit of explanation, explaining it right now when we're doing protein synthesis. So, before we start, let me ask you this. How do you think messenger RNA is formed? And how do you think protein synthesis is initiated in the body? And how does DNA control protein synthesis? Okay, so as I was saying, I'm going to explain protein synthesis, but in a very different way. Let's start using the book, how to explain it in the book, and then I'm going to explain it using the way that I have been explaining it for the past 3 years. Okay, so what happens during protein synthesis? The first part of protein synthesis, it's almost like DNA replication. So what happens? The first thing that happens is that, since we do know that DNA, this is the part where I explain how DNA controls protein synthesis. So the first part is DNA, as you know, is a double helix. So what it does, it unwinds. When it unwinds, and then it will unzip. When it unzips, and then when I'm talking about when it will unwind, and then when it has unwinded, it will also do the breaking of bonds. The bonds will break. When the bonds have broken, it will then unzip, which means it separates. So from a helical shape to a leather-like shape to a single strand like this. So when this happens, this means it has separated. When this happens, since we know that DNA has 2 strands, only one strand, since we are talking about protein synthesis, only one strand of the DNA acts as a template. So when we are talking about a template, it's like, how can I say what's a template? A template, I could most probably say it's a blueprint of some sort. So when we are saying it acts as a template, so as the DNA, this is your DNA strand, the one that is acting as a template, and then these are nitrogenous bases. Remember, this is what? A DNA strand. This is your DNA strand. Okay. Remember that I said when we are talking about RNA, we do not have thymine, but we have what? The uracil. So whenever you see a structure or a strand that has a U in it, know that U, when it has a U, that means it's what? It's RNA. But whenever you see a structure with a T, that means it's what? It is DNA. So when we are talking about DNA, so now this is the DNA, and then the DNA strand that is acting as a template for the creation of RNA. Which RNA? I will explain it to you now. So we all know that adenine, when it hits with DNA, it pairs with thymine. But since we are making a RNA strand, this is the messenger RNA. It's a messenger RNA. It's what, when we are talking about messenger, like on your phone, messenger, what does it do? It takes messages from one part to the next, right? So when that happens, so does the mRNA. It's a messenger. It takes a code from DNA and then goes to the ribosome where it's the site of protein synthesis, and then it translates the code. So this is what happened. Adenine, because if this was a DNA strand, then we would have put what? Then we would have put T for thymine. But then now we are no longer going to put T, we are going to put a U. Because we are doing what now? We are dealing with RNA. And then T will still pair with A, and then U, and then we have C, and then we have C, and then we have U, and then we have G. This is your messenger RNA. So we are done with DNA. We are done with DNA. Since we are done with DNA, I will give you a short moment just to look at that. Whilst I look at something. When we are talking about the triplet bases, these are the triplet bases. We call them what? Codons. The triplet bases for messenger RNA are called codons. And then the triplet bases for transfer RNA are called anticodons. So as to remember, anticodons, so you can always remember this, you cannot forget this. Anticodons have a T for transfer RNA. And then we call them the triplet bases. Anticodons, they have a T. So when they have a T, what does that tell you? They are for transfer RNA. And then you have codons. They do not have a T, they do not have an R, they do not have an N. So they are what? They are for, just remember, codons are for mRNA, which is messenger RNA. Another thing that I've seen over the years that I've been tutoring, most of my pupils make a mistake. Whenever you're writing the acronym for RNA, you never put M as a capital letter as RNA. But what you do, M is a small letter, a lowercase letter, and then RNA. Again, with R for ribosome, you have like this. T for transfer RNA, they are all lowercase letters, followed by the capital letter acronym, which is for ribonucleic acid. Okay, so since this is done, we have our messenger RNA. What does it do now, since it has been given there? So, this is the code. Remember I said DNA controls protein synthesis. This is the code. Since it's adenine, thymine, and adenine, and then it's creating or forming messenger RNA, it's telling the messenger RNA that, okay, these are my triplet bases. Then they should pair like this, so that this is the code that is given by DNA to messenger RNA for the formation of the protein. So DNA gives the code. The first stage of protein synthesis is called transcription. Transcription. When you're talking about transcription, you are transcribing something. It's like a doctor giving you a letter to the pharmacy. So I'm going to tell you that go to the doctor and go to the pharmacy and pick Panadol for your headache. I am prescribing something for you. Same does DNA. What it does, it gives a code for messenger RNA to tell messenger RNA to go and tell the tRNA that these are the amino acids that I need. So this is the protein that is going to be formed in the egg. So, and then we also have what? We also have the movement of messenger RNA out of the nucleus. So when the messenger RNA has been created, what then happens is that since we know that the nucleus, I'm not going to be drawing, forgive me. So when we are looking at the nucleus has nuclear pores like this. So the messenger RNA moves out through the nuclear pores and goes to the ribosome, which is the site of protein synthesis. Okay. Okay, so when we are talking about the movement of messenger RNA, it moves through the nuclear pores. Since we know that the nucleus has pores, so it moves out of the nucleus, out through the nuclear pore into the cytoplasm. And when it is in the cytoplasm, it goes and attaches itself where? In the ribosome. So let me do a quick recap. We have a cell, and then within the cell we have the cytoplasm, and then within the cytoplasm, in the cytoplasm we have organelles suspended in the cytoplasm, and then one of those organelles is what is called a ribosome, which is the site of protein synthesis. So now, when the messenger RNA has attached itself into the ribosome, then it starts another process, which is the third part. In some books, it is differentiated into transcription, movement of the messenger RNA, and then translation. In some books, there is only two, which is transcription and translation. So whichever book that you are using, it is correct. It is not wrong, but make sure that you do not have one source of information. Look at different books. And make sure that when you are looking at those books, do not only rely on me tutoring you, but also be in contact with your teacher. Okay. So translation, when we are talking about translation, it's in simple English. When we are talking about translation, it's whereby I'm taking something that I do not understand and someone is converting it into something that I can understand. Like, I am talking to my friend who is maybe from Mozambique, and they are speaking a language that I do not understand. So I need someone who's going to translate that, so that when they talk to me or I talk to them, we understand each other. Same as translation. So when the messenger RNA has attached itself on the ribosome, what then happens? So according to the codons, according to these codons, they give out, it's like, it's like a beacon, or I don't know what to call this thing. Yeah, it's like a beacon. When you are using a microchip to track something, and then it's emitting sounds or waves, so that you are able to track where it is. So this is the same thing that occurs with messenger RNA. What it does when it has attached itself on the ribosome, it will give out a code. It translates, it gives out a code, a sound of sort of that is heard by the transfer RNA, telling it that these are my triplet bases. Since these are the triplet bases, UAU, uracil, adenine, and uracil. So I need an amino acid that I need a transfer RNA that will be able to match these. Whenever a transfer RNA comes, it's like a car and a trailer. The transfer RNA is the transfer RNA, but then the trailer is what? Is the amino acids. So the transfer RNA are transferring, as the name says, the transfer RNAs are transferring the amino acids to the ribosome, where protein synthesis will occur. And then as they come together and join, what happens? It's one amino acid after the other, after the other. And when they are joined together by what? By peptide bonds. But when they are many bonds, we call them what? Polypeptide bond. Polypeptide or peptide. Whichever one makes you happy and you understand more. Okay. So when these amino acids have come, they form a chain, and then that chain, the smallest chain is made of 50 amino acids, if I am correct. If I remember properly, yes, it is made of 50 amino acids. So when these codons translate a code, what happens? The transfer RNA comes and attaches itself on the messenger RNA in the ribosome, and then your proteins are formed. Because the transfer RNA, as I said, it's a car, and then the amino acids are a trailer. And then behind the trailer, all these transfer RNAs come and then they join together. All these cars in their trailers, they join together to form what? A protein. Now let me explain this in a way that you may understand, which is simple. Here's my phone. We are used to using social media every time, every day. Right now, just check if I have any message on WhatsApp. So let's explain protein synthesis using WhatsApp, something that you do every day, using Snapchat, using Twitter, replying to someone, to a friend, a message, whatever. So what happens? Whenever you are, when you want to communicate with someone, you pick up your phone, and then when you're picking up your phone, you start typing a message. When you type a message, that is, you are the DNA strand, you are giving a code to your phone. So as you are giving a code to that person, as you are typing, that message that is being written there, that is your messenger RNA. So you type. I love you, I miss you, I do whatever I want to do, whatever. I want a party for Christmas. The message that you are typing on your phone is what? Is the messenger RNA. You are forming the messenger RNA since you are the DNA strand. And then when you are done typing that message, what happens? You press send. When you press send, it's whereby this messenger RNA moves out of the nuclear core and goes and attaches itself onto your friend's phone, or your father, your mother, or whoever, your sister, or whoever it is that you are trying to talk to. And then when you do that, when they get the message, that means it's the messenger RNA has attached itself onto the ribosome. So when it has attached itself, what happens? The first thing that happens, an alarm goes off, like a notification sound goes off. When the notification sound goes off, it's telling the other person that there is a message, come and read it. So when that message comes, then, oh, I have a message. When they are reading the message, like right now, I'm going to read this book, pretend it's my phone. I'm going to read it. The process of protein synthesis. Protein synthesis takes place in 3 stages. So when I am reading, it's whereby I am taking in the code, translation of text. I am taking the code from the text, and then I am reading it. And then as I reply, okay, he's saying this, okay, she's saying this. As I reply, I am then, what am I doing? I am giving off my amino acids. I'm giving transfer RNA. I'm typing. As soon as I send reply, that means the process of protein synthesis has been complete, because as I send a reply, that means transfer RNA has actually been able to identify what the correct sequence of anticodons need to go there. What is the appropriate response that needs to go there, so that they are able, that person is able to get what they want. So it's easy as that. It's like chatting every day. That is your protein synthesis. You send a message, you receive a message. You send a message, you receive a message. That is how it happens. Messenger RNA is formed, and then transfer RNA, brings in the amino acids, and then everyone is happy with the formed proteins. And I hope you have a very good day. That's it for today, and then tomorrow we are going to talk about meiosis, and we are going to do meiosis 1 and meiosis 2 tomorrow, and do not a deep revision again. I will try. I talk too much. I love talking. I'm a teacher after all. So thank you for your time. I hope you understood everything. If you have any questions, feel free to use the number on the screen.